So this lecture is all about vision, how your eye works to transfer information from the outside world in terms of light energy into senses and your perception of that information. Now, the eye has evolved separately in numerous organisms because it's extremely important to navigate your world visually. So this lecture is all about how that process works. There's also been a lot of really interesting advances in vision. We're going to talk a little bit about cochlear implants for hearing, but there's also been some work on such things as the artificial retina or the bionic eye, and I think we're going to see that as a big part of the future. Okay, so there's the eyeball, the basic eyeball. And I'd like you to know a little anatomy about that. Okay, so starting in the left, here you have the cornea, and that helps to bend the light. The cornea bends light. You have some liquid in here, and that's called aqueous humor. Humor is a liquid. The lens then bends the light again to accommodate, we use the word accommodate, that light information on the back of the eye, the retina. Now, there's some muscles here, ciliary muscles, that bend that lens to enable a person to focus that light right on the back of the retina. Sometimes an eyeball is a little long or a little short, and so we have to have glasses or contact lenses to help in the accommodation. The older you are, the more rings you have in your lenses, the harder it is for it to bend. So oftentimes older people like myself have to take a piece of paper and put it out just a little bit to be able to read what we're looking at. Now, what we're going to talk about here is a word called transduction. And transduction is about taking light energy and transferring into something the brain can use. And what the brain can use is simply action potentials. It's going to transduce it at the retina and then send signals back through the optic nerve. Those are those signals, just like action potentials, which we've talked about. Here's just another picture. Now, right in the back of the eye, where you really focus, you have an area called the fovea. And the fovea has a lot of densely packed, color-sensitive cells that transduce detail and color. And we'll talk about that in just a moment. But the light is going to actually transfer through the retina, this is in the back of the eye, through cells that don't transduce light energy. These are the ganglion cells and the bipolar cells, the intermediate cells, and finally we get back to these cones and rods. Here's another picture of the retina. See the light transfer through the ganglion cells? The ganglion cells are what are going to be used to send signals back to the back of the brain, back into the thalamus, and eventually back to the back of the brain. They aren't going to transduce light. These intermediate cells, and I don't need you to know all of them, they're just called intermediate cells. They don't transduce light energy. It's these cells back here, the cones and the rods. I bet you've heard of cones and rods before. Rods tend to deal with black and white. Cones tend to deal with color and detail. So that is kind of an image of the retina. So the first thing that light really hits in the retina are ganglion cells, but it really doesn't affect them. These are going to transduce, and then they're going to send the signals the other way. There's the retina again. This is actually, I guess, a flip version of what you saw on the last slide. The light is coming in from the right. It's transduced here at the cones and rods, changing action potentials, which are then going to affect these intermediate cells and the ganglion cells, and the ganglion cells are going to send their signals back to the thalamus through the optic nerve. The axon bundles of the ganglion cells are optic nerves. That's a nice microscopic vision of the rods and cones. You can see the rods kind of look like rods and the cones kind of look like cones. It's nice that something is actually named properly. So let's go over a little bit about rods and cones. We talk about rods as having or responsible for scotopic vision. What is scotopic vision? That's night vision. That is achromatic, not light and dark, not color, but it tends to deal with light and dark. Light and dark. Also really good for movement. These rods tend to be in your peripheral vision. Not exactly what you're focusing on, but a little to the right, a little bit to the left. But they're very good about sudden movements. This is very important evolutionarily, because if something is jumping out of the bushes to attack you, you have to be very sensitive to that movement, and rods are very good at that. About 130 million rods in your eyes. Cones are for photopic vision, daytime vision, light color, color vision, foveal vision. And these are the ones that are sensitive to different wavelengths of light. Light are broken up into wavelengths. And so we have red cones and we have blue-yellow cones, all sensitive to different wavelengths of light and react to those wavelengths. We call that foveal vision because most of the cones are right in that spot of your eye that you're focusing on. About 7 million cones. Now, if we were to look at the fovea, right where you're focusing, we have an optic disc here where we actually can't see anything. We kind of make up for that. We don't really see those black spots. But you can see the high level or number or density of cones right here in the fovea and a few in the periphery. And the rods, most of them are in the periphery, 10, 30, 40, 60% a degree off of your focus vision. Okay, now back to transduction. Now we transduce a lot of things. We transduce light into action potentials. We transduce air sound waves into action potentials because that's all we can deal with. We can only deal with action potentials. That's how we decode the world. So we have to transduce the light energy into something that our brains can handle, and that is rates of action potentials. So we're going to transduce light into action potentials. We have to have a mechanism to do that, and do that in a very specific way. So let's talk about that one thing of transduction. Let's take a look at a rod here. A rod is a modified neuron, and it has the nucleus and it has the mitochondria and everything, and it also has these stacks here. These stacks which have chemicals in them. And one of the chemicals that it has that is responsible for transduction, that is sensitive to light, is a molecule called rhodopsin, which is made of opsin and retinol. And this is a molecule that changes when light strikes it. It's very rare, in terms of our body, that light affects a molecule in this way. And what we're going to do is we're going to hit that with light, change its shape, and that's going to cause a chain reaction which will ultimately affect action potentials. So here's kind of a complicated diagram of it, but it's my best shot at animating what is happening. So up here is the discs, the outer discs in a rod, and I have that as being right in here, right in this section. And here is a rhodopsin molecule in this lipid bilayer in the disc. And here is retinol. And what happens, though, on the outer part of the membrane, which I kind of diagram over here, it's very hard to diagram this, you have a constant flow of sodium. In the dark, when the rod is not being hit by light, you have this constant flow of sodium coming in, a constant depolarization. Remember, sodium causes depolarization. Now, here is a sodium channel, and it's being held open by a molecule called cyclic GMP, specific type, and it's holding it open. And sodium is constantly rushing in, constantly depolarizing this membrane and affecting action potentials. They actually have a name for it. They call this the dark current, the current of sodium flowing in the dark current. But if we're in the light, if this rod is hit by light, a chain reaction happens. So let's see if we can follow what happens. So keep an eye on this rhodopsin molecule and this retinol in here with a certain shape. Here comes a photon of light, and it strikes that retinol, and it changes its shape. Light changes its shape. We call that a photopigment changes. Now that change in shape causes a chain reaction. Now, the specifics of that chain reaction, I don't think you need to know, but it's eventually going to affect the cyclic GMP and destroy it or remove it temporarily. If cyclic GMP is removed, the sodium channel snaps shut. Remember, cyclic GMP is what's holding that open. Now, sodium isn't coming in, and we've changed the polarity of that membrane, ultimately changing how that neuron fires. It's all about firing rate, and it's all about membranes either being depolarized or not depolarized and affecting that rate. Now, if no sodium is coming in, then we're not going to release as many neurotransmitters here, all because of that light. So let's watch it one more time. Sodium is coming in the dark. A photon of light comes in, changes the retinol molecule, changes its shape. This ultimately causes a chain reaction which gets rid of cyclic GMP, which is opening the sodium channel. Sodium channels close, sodium can't get in, we've changed the polarity. That's all we've done. That's a transduction from a light to affecting action potentials. Here's another diagram of what I just said without my great animation, but you can see here, these are the sodium channels coming in, the dark current, okay? Held open by a cyclic GMP, holds it open. When light strikes a rhodopsin molecule in the membrane of these stacks here, it is going to affect, through this chain reaction, that cyclic GMP. Sodium can't get in anymore. We've changed the polarity of the membrane. We've changed the firing rate, and that's all we've got. We've only got rates of firing as our code. Light has changed that rate, and we have a different signal. We have a signal where the brain says, there must be light striking this. Of course, you have all these millions of rods and cones, and it can get very complex. But we saw transduction. So let's go over transduction as a summary. In the dark, opsin, retinol, rhodopsin are bound together. They have a certain shape. Rod membrane is depolarized slightly because of the dark current, sodium coming in, sodium coming in. Why does sodium come in? Because cyclic GMP is holding that sodium channel open in the dark. But now it's in the light. Now that rod, that one rod, is hit by light, a photon of light. Now, that is going to affect the photopigment, going to affect rhodopsin, going to affect the shape, causing a reduction in the cyclic GMP that's holding the sodium channel open, and the rod hyperpolarizes because sodium channel is shut, sodium can't get in, can't depolarize the cell. We've changed the rate of firing of that neuron. Okay, so we've changed the rate of firing, which will ultimately affect intermediate cells, will ultimately affect ganglion cells. The ganglion cells create that signal, that nerve signal goes down, crosses over. We're going to look at that visual pathway, how it crosses over. We call that the optic chiasm. You can actually see that in the ventral portion of a brain. There's a little portion right above the chiasm called the suprachiasmatic nucleus. That will become important later when we talk about things like emotions. And finally, the signal gets back to the thalamus, an area of the thalamus called the lateral geniculate nucleus of the thalamus. I just like to call it the LGN. The LGN. It's a big lateral geniculate nucleus, a very long word. And also, we'll get a signal to the superior colliculus, which we'll talk about in another lecture. And finally, back to the back of the brain, to the occipital lobe, to the primary visual cortex for the process of assembling that image. Now, what happens is we get a crossing over of some information and a not crossing over of others. It's not like the right eye, all the information from the right eye goes to the left side of the brain. We call that a contralateral. Some of the information from the right eye goes contralateral. Some of that information, which is out here on this part of the eye, this is the nose in here, stays ipsilateral. So the right eyeball is being decoded by both the left and right part of the primary visual cortex. It's the same for the left eye. We talk about fields. All the stuff over here is being processed by the left side, and all the stuff on the left is being processed by the right. Okay? Contralateral, we get a contralateral from the part nearest the nose, and an ipsilateral, staying on the same side, from the part nearest the temple. That's just another way of looking at it right there. Those are the fields that I was talking about. And I think this image helps to show you what part of the retina is going across here at the LGN. And this part, all the stuff in green, stays ipsilateral. This goes contralateral. Okay? So we get this crossing over of some information and the staying on the same side of other information. Finally, that visual pathway after the optic chiasm goes to the LGN, the lateral geniculate nucleus of the thalamus. And it's actually broken up because a lot of it, we call it the magnocellular layer. I don't think you need to know that, but that's getting scotopic vision. That's getting a lot of information about movement, about light and dark from the rods. And then we have over here, we call that the parvocellular layer of the thalamus. That's getting a lot of foveal vision, a lot of scotopic, photopic vision, a lot of vision from the cones. Just another picture. You can see the LGN over here. Remember, the thalamus is kind of a way station. It takes a lot of sensory information, and it takes it and sends it from the point where we have transduction in the ear and the eye and the touch, and sends it over to be decoded in different parts of the neocortex. There it is, LGN. There's just a sagittal section of the brain. You can see the thalamus there. Finally, that information from the LGN is going to send information back to different parts of the primary visual cortex. And a lot of times it's coded by V's, V1, V2, V3, V4, all those kinds of things. And maybe you learned that in a different class. But we're going to learn about some cells back in here and how they decode. A lot of that information comes to V1, but maybe we might be decoding some functions of movement or color in some of the other V's. There's that primary visual cortex as seen by sort of overlaying what probably is a PET scan. There's just the ventral surface of the brain, so you can see the optic chiasm, the optic nerve coming from the ganglion cells in the retina, coming back to the LGN, and then the LGN fanning out and sending those signals to a lot of parts of the primary visual cortex, and this is where we're going to build up some images. Now, one thing that I'd like you to have just a basic understanding of is the fact that we have these kind of fields, that the retina and the LGN and the primary visual cortex, it really deals with fields that are kind of off and on fields. In the retina, these fields are circular. So if you have these intermediate cells here, they might be responsible for a field of cones. And on those cones is kind of a donut shape. That's the field they're responsible for, is a circle. All the cones send their information eventually to these intermediate cells and eventually to ganglion cells. And there's the fields on the retina and the LGN are circular, and we call them on center and off center. It's a very simple concept. It's the idea that a neuron that's maybe an intermediate cell, it fires the most. If it's an on-center cell, it fires the most when light strikes the middle. When light strikes its peripheral circle, it fires less. Remember, that's the only codes we have, is firing a lot, firing a little. So some of the cells fire a lot when the middle of their field, the cells they're responsible for, is hit by light. And some are off-center. They fire the most when the outer part, the periphery of their field, the cells that they're responsible for, is hit by light compared to the inside. And we have a lot of overlapping fields, so you have a lot of on and off center. The point here, and you can maybe get this in another class on senses and perception, but the point here is we're trying to highlight light and dark spots in our visual field, where light and dark come together. Edges and lines, we want to highlight that. It's the beginning process of building up a visual image. So if I had a cell, like an intermediate cell or a ganglion cell, for example, and its field was a bunch of photoreceptor cells, like rods and cones, and a lot of those neurons, those rods and cones, eventually signal the ganglion cell, that's their field. If we shine a light right in the middle of the field and we're recording from the ganglion cell, like some very famous researchers had done, the cell fires a lot. However, if you move that light and fire and shine it on the periphery of the field, the cells that the ganglion cells are responsible for, it fires very little. Up and down, that's an on center. And so you can imagine what an off center would do. It would fire a lot more when it's the periphery that it's attached to. Okay, so the researchers that did all this work are actually won the Nobel Prize. They're Hubel and Wiesel, and they won the Nobel Prize in 1981, and they're the ones who discovered these receptive fields both in the retina and in the primary visual cortex. What they found was that cells in the retina respond in the way I just described, these on-center, off-center fields. But when recording from cells from a cat in the primary visual cortex, they found the receptive fields not to be circular, but to be more rectangular. And if the center of the field is hit, it might be an on-center or off-center, they have different firing rates. Remember, it's all about firing rates. It always comes down to firing rates. So neurons in the primary visual cortex, sometimes we call the striate cortex, they become very sensitive. They represent part of the retina. They're getting signals through the LGN, but they really are representing part of the retina. We actually call that retinotopic. In other words, it's kind of the back of the brain is kind of a map of the retina. They become specialized. They become specialized cells. And they only like certain types of stimulation on their field. Some of them are very sensitive to movement across their field. Some are very sensitive to lines of specific orientation, a specific size, a specific direction. And we begin to have a lot of cells that begin to send signals further on about what's happening on their field. So Hubel and Wiesel were recording from these simple cells, these single cells in the primary visual cortex. And they were finding that these stacks of cells were kind of organized in a very specific way. Imagine these are cells that you would find in the primary visual cortex. Each box is a cell. But these cells are all responsible for, let's say, the same field, a map of the retina, but they fire at different rates. So if there's a completely vertical line on that part of the retina at that very moment, these cells here fire a lot, but these don't fire hardly at all. But what if that line changes? That changed because the eyes are moving all the time, and that line changes, and suddenly the line is more horizontal. Well, now these cells in the middle are firing a lot, and these at the end are firing a little. And they're just all singing, they're all setting their action potentials at different rates further along the process. Imagine millions and millions and millions of these cells, all with different receptive fields. Well, that's kind of a complicated version of it, but you can see the different cells being responsible for different orientations coming from the left and the right eyeball. Okay? So we call those cells that are very specific to orientation, we call those simple cells. So the simple cells like lines of very specific orientation, vertical, horizontal, between vertical and horizontal, and that's how they fire. They send signals to complex cells in the primary visual cortex. And these cells are also getting information from the LGN. And they like cells of specific orientation, but they also like specific orientation of lines that move in a very specific direction. So it has to be a vertical line that moves in a specific direction. They have a much larger field, receptive field. So they react to movement in specific directions. I hope you're seeing how we're building things up. The lines and the edges of your visual field are accentuated at the retina. The different parts of the primary visual cortex in the back of the head are getting signals about lines of specific orientation and edges and movement. And what it does is it helps to fill in the gaps. What we have in this process is really the brain begins to work in what is known as a parallel process. The simple cells, the complex cells, other cells begin to say, this is what I think you ought to be seeing. They're working back and forth. They're exciting and inhibiting back and forth to help you see what the brain thinks you ought to be seeing. It is taking into account context. It's taking into account memory. And so you create these nice, fun optical illusions that demonstrate this. I mean, here's an image. There isn't a white triangle there. Your brain wants to say there's a white triangle there because it fits. There's enough information. And you can actually see lines kind of, unless you stare right at it. If you're just looking at the whole picture, lines here and here kind of are created. And this is a process of the simple cells and the complex cells and other cells building up that information and sending it to the temporal lobe here. And the ventral surface of the temporal lobe, this place called FG, is called the fusiform gyrus. And we're beginning to build things up because there are cells there that are not sensitive to lines of specific orientation, of movement, of angles, but maybe of kind of objects. They only fire with certain objects. So you might have cells that fire, a lot of cells that fire when they see certain types of faces or hands or shapes. And they're getting information. Now this is key. They're getting information from simple cells, complex cells, and they're sending information back to try to modify. Use your memory to think about what you think you see. Here's an example of face cells. We have face cells in the fusiform gyrus. These cells respond to faces. So if you're recording from these like they did with these specific types of a primate monkey, they're firing the most here. But if you start rearranging the face, changing the face, you begin to get lower firing rates from the face cell. You show them a hand, that cell's not firing at all. And you might even have cells, face cells, that respond only to faces of specific orientation. So here's a cell that they're recording from that really likes a full front direct view of another monkey. Well, if you begin to change that monkey's orientation that they're seeing, the cell gets less and less firing rate. And if you remove the eyes, it's just not firing as much. These are face cells. You know, there's even thoughts that we have grandmother cells, cells that fire a lot when you see a very familiar face, like your grandmother. This is all of that building up. This is all that building up, starting with lines and edges of specific orientation and building that image. But that's a serial process going from the retina to simple cells to complex cells to face cells. But you also have a parallel process where all these cells are kind of modifying, what you think you see. Have you ever seen a friend across a parking lot and you're sure that that's your friend and you see them and they walk towards you and you wave at them and all of a sudden, at a moment where they're close enough, it's not your friend anymore. You mistook that face, which was very similar. The brain almost goes close enough. I'm going to make you think. I'm going to make you see. I'm going to make you perceive your friend. Here's kind of an example. I love this painting by Bev Doodle. I think it's called The Forest Have Eyes or something. But if you look at this image, it's a horse and a rider on a horse, and you can see that. But you can also begin to see faces in this image. Your brain starts bringing together enough information for faces, and you begin to see faces in the forest because your mind is set and wants to see faces. We see faces in the strangest things. Here's a couple of optical illusions that I like that help to indicate to you that what the senses are telling your brain can be very different from what you perceive because it takes into account context. It takes into account memory. Take these two circles here. These two circles, one looks yellow, one looks green. At least they do to me. But the brain is saying, well, this is in the shadow, and this is in the bright light. So something must have changed, and I will modify what you see because I know that this is in the shadow and this is in bright light. But the truth is that these two circles are exactly the same color, exactly in every way. This is the same circle. And I can even prove it to you. I'm going to take that circle and I'm just going to move it. I didn't change it. I didn't change that color. It stayed the same color it used to be. But the context makes your brain modify it. So what you see is oftentimes different from what you perceive. See, if I even take all that context out, I'm just going to remove the context and keep the circles there. You can see that the circles are in fact the same. I'm just removing the background. Context plays a big role in perception. It has a lot to do with the parallel processing. Here's another optical illusion. Two surfaces, but it depends on the context. These are the same exact image, the same exact parallelogram, because if I just take it and move it over there, you can see that they're the same. But if I had context, this one over here is shorter and much fatter this way, and this is much longer than this is. But context shapes what you see. So let's go through a visual system kind of just basic summary. You should know a little bit about transduction, that process of taking light energy and transforming it into a neural signal that the brain can deal with. This happens at the retina. You should know a little bit about the anatomy of the eye, how that light gets there, and how it's transduced. We get a little bit of modification already of that information by these center surround fields. These happen at the intermediate cells. These happen at the ganglion cells. We're already modifying that image to accentuate some edges and lines. Signals goes back to the LGN, and we're going to talk about the LGN in future lectures. We'll talk about it when we talk about emotions and a few other things. But that's kind of a way station for information, and that the ganglion cells from the retina go to the LGN. The information from the LGN then goes back to the primary visual cortex. And then we have the primary visual cortex builds up that images, lines and edges and angles and motion, and it does it both in one direction and in a parallel back and forth to modify and to keep in mind that context and memory play a big role in what you actually perceive.